Okay. Okay, properties of muscular tissue, electrical excitability. So the muscles are stimulated to contract um, by um, mostly by nerve impulses. So if I decide that I'm going to bend my elbow, my brain sends a nerve impulse to these muscles, and then that excites the muscles. So it's like the muscle cells have their own kind of nerve impulse that causes them to contract. See how exactly how that happens. Contractility, which means they can get shorter, they can contract. And extensibility, which means that when they can contract, but they can also stretch back out to their stretch length. And elasticity, the muscles are stretchy. Three types of muscular tissue. We already learned this back in chapter 4, I think. Skeletal, cardiac, and smooth. And we learned some of the basic differences. Skeletal muscle is attached to the bones. That's why it's called skeletal, of course. So this provides movement of the skeleton. That is flexing, extending, twisting joints, generating heat. Anytime a muscle contracts, it produces some heat as a byproduct. You get hot when you're exercising or working hard. Maintaining posture. So even if I'm not necessarily moving, but just standing still like this, it's muscles contracting that are allowing me to do this. So muscles, even though they're not causing any movement, they're contracting, keeping my body in this position. Prevent skeletal structure damage. Your muscles provide a layer of cushioning over your skeleton. Stabilize joints. For example, if I'm lifting something that's kind of heavy with my right arm, even though it's the muscles of my arm that are doing this, my whole body has to kind of stiffen up, or otherwise I would lean over or collapse. So even though it's my arm is doing this, my whole upper body and the shoulder has to be stabilized. The muscles from my chest and shoulders all tighten up. Shield internal organs. Many of our internal organs are protected by bone, the cranium and the thoracic cage here, but the abdominal organs, stomach and liver and intestines here, there's no bone really covering them. But we have this thick layer of muscle that's pretty protective of those organs. The appearance of skeletal muscle is, under a microscope, is striated, so we see those lines, those faint lines across, called striations. The fibers, remember the term fiber means cell. If we're talking about muscle, so a muscle cell is called a muscle fiber. In skeletal muscle, the muscle fibers or cells are parallel, and they're all running in the same direction. So this muscle here, a bundle of these muscle fibers that are long like spaghetti, all in a parallel bundle. And each muscle fiber or cell has many nuclei. You can see those dark spots. That's because each one of them started out as many cells in a row, and they all fused together into one long cell. Then all the nuclei are still there, the original else. This is the only kind of muscle that is voluntarily controlled. That means you consciously decide that you're going to contract a muscle and your brain makes it happen. Your brain sends nerve impulses to that muscle through a nerve impulses. Cardiac muscle only occurs in the heart. So this is what contracts to pump the blood out of the heart and through your circulatory system. It also appears striated. It's hard to see, the lines are very faint, but there are lines across those muscle fibers. Just one nucleus per fiber or per cell, involuntarily controlled, so you can't consciously decide to speed up your heart or slow it down or stop it. Does it by itself. The autorhythmic. Auto means self. Rhythmic means in a regular, timely manner. So your heart beats by itself. It has its own pace in it. It doesn't need the brain telling it to contract every time. It does it by itself. It's its own pacemaker, and it beats at a regular rhythm. Even if a heart is removed from a person, so it's no longer connected to the nervous system at all, it'll keep beating. But when it's in your body, your nervous system can speed it up or slow it down, and hormones can speed it up or slow it down. The smooth muscle. In the many internal organs have smooth muscle. Blood vessels are surrounded by smooth muscle that make them constrict and dilate. Functions peristalsis, peristalsis is a wave of contraction along a tube. That's how food is moved through your intestine. That's how you swallow food. That swallowing is just a wave of contraction along your esophagus. Contracts right behind the food, and that wave of contraction just moves down your throat, pushing the food along in front of it until it gets stomach. Vasoconstriction. Vaso refers to blood vessels, so this just means blood vessels constricting, getting narrower to reduce the blood flow. Lots of other functions. The little tiny muscles surrounding the pupil in your eye, muscles in the iris, and that's what opens and closes the pupil. When you're in bright light, the pupil gets smaller. When you're in dim light, it gets bigger because of those smooth muscles we learned about. The erector pili muscles, remember? Muscles that are attached to the hair follicles, but smooth muscle, involuntary, causes goosebumps and causes hair to stand up. So involuntary control, of course, no striations. That's why it's called smooth, because it doesn't have striations. Just one nucleus per fiber. Activated. Some are activated by stretching. Some are by hormones. And some are by the autonomic nervous system. Autonomic part of the nervous system means the involuntary part, the automatic. I always think autonomic, automatic. It happens without us consciously making it happen. Okay, we'll learn more about the cardiac muscle when we do a whole chapter on the heart later. So the same information here. The shape of these muscle fibers is different in the 3 different types. In skeletal muscle, they're long like spaghetti. In the cardiac muscle, they're shorter and they're branched. each fiber is branching, so it could, one muscle fiber could look um, like this. Branched. Sometimes they're branched at both ends. That would be one muscle fiber of cardiac muscle. And so these, each end, the end would be connected to the next one. And where they're connected here, these are called intercalated discs. Intercalated disc. They just look like dark lines labeled on there. Only cardiac muscle has intercalated discs. A smooth muscle, hard to tell how they're shaped, but they're kind of long and tapered or pointed on the ends. The smooth muscle. Okay, is that same features compared in a table. Okay, skeletal muscle, we don't need to learn about this. Just like I said, each muscle fiber is formed by many smaller cells fusing together. Within a muscle, there are connective tissue coverings of the entire muscle, and that's called an epimysium. So this whole biceps muscle has a thin covering of connective tissue over it. And then within that muscle, there are bundles of muscle fibers or muscle cells. And those bundles are called fascicles, and each fascicle has a layer of connective tissue covering it, called a perimysium. And then each individual muscle fiber or cell has a connective tissue fiber called an endomysium. And like it says here, these at the end of a muscle, these connective tissue coverings, they just continue on and become the tendons, which then connect to the bone. So in for example, this biceps muscle here, it has a covering over the whole muscle, and then within the covering of the bundles within. And all those coverings, they just continue on past the muscle and form a rope, which is a tendon that then attaches to the bone. Same thing at the other end. This word aponeurosis is just a big flat tendon. So instead of, most tendons are like ropes, aponeurosis is a big flat tendon. So think of this pectoral muscle here. At this end, the shoulder end, that there's just a regular tendon that pectoralis muscle attaches to the shoulder, as long as it's the shoulder. But at this end, that muscle just spread out, and it attaches all along the sternum. And so there's a thin flat tendon that attaches this muscle to the sternum, and that's called an aponeurosis. This word fascia is in some parts of the body. There's these bands of fibrous connective tissue that it's called a fascia that surrounds some muscles and organs of the body. I mentioned when we were talking about the feet, the bottom of the foot. There's that tissue that goes from the heel to the bottom of the ball of the foot, the main arch, the arch of the foot. That's called the plantar fascia. It's one of the bands of tissue. Okay, so here's a diagram showing the structure of a muscle. Up at the top here, that's a whole muscle, so that could be the biceps muscle here in the front of the upper arm. And so there's the tendon at the left end where it attached to the bone. But within that muscle, there are these bundles of muscle fibers, muscle cells. Those bundles are called fascicles. And within each fascicle, there's a bunch of muscle fibers or cells. So each one of those oblong structures is just a cross section cut through one of the muscle fibers or cells within that fascicle. Here we see an enlarged fascicle, and so this is one muscle fiber, one muscle cell, and these dots here are all cuts, section cuts through these myofibrils. Each myofibril is a bundle of proteins, long protein molecules that run through there. And it's the myofibrils that can get shorter. Each muscle fiber has all these myofibrils, and these myofibrils can get shorter, and that causes a whole muscle to get shorter. And we'll have to learn how a myofibril shortens itself. Again, an even bigger view of a muscle fiber with these long myofibrils made up of long protein molecules. These are it's labeled these mitochondria in there. Remember, mitochondria are the organelles where nutrient molecules like sugars are broken down and the energy is released and stored as ATP for the cell to use that energy later. Muscle fibers have a lot of mitochondria because muscle fibers use so much energy. It's a lot of energy just to do this. The muscle, contract the muscle. Also labeled there sarcolemma. That's just the plasma membrane surrounding that whole cell. The plasma membrane of a muscle fiber has its own name, and it's called a sarcolemma. That's all it is, just the plasma membrane. So here's this myofibril. One of these myofibrils is enlarged here, or a section of a myofibril. But this actually runs the whole length of the muscle cell. So this myofibril is what can get shorter. All these myofibrils will get shorter at the same time, and that'll cause muscle cell to get shorter. And so when the muscle cells get shorter, the whole muscle gets shorter. That's how we flex our joints. A myofibril is made up of a bunch of sections called sarcomeres. So here's one sarcomere from here to here. And then there will be another sarcomere here, another one here, another one here. So there's many sarcomeres along the myofibril that runs the length of that muscle cell. Each sarcomere, this is what gets shorter, the sarcomere. So this is called a Z disc, like a letter Z. That's just where one sarcomere is connected to the next one, the Z disc. And these lines here are protein molecules. The dark ones, these dark thicker ones are called thick filaments. Thick filaments, and they're made up of the protein myosin. These thinner, lighter colored ones are called the thin filaments, and they're made up of the protein actin. And you can see that the actin, the thin filaments, overlap the thick filaments. Here's the thick filament, and the thin filament overlaps it here. And how this thing gets shorter, how this sarcomere gets shorter, is that these thin filaments and the thick filaments will just overlap more and more. And this whole the Z disc will get closer together, and the sarcomere gets shorter. So if this sarcomere gets shorter, and this one gets shorter, and this one gets shorter, they're all getting shorter. The whole myofibril gets shorter. And this muscle fiber is, I don't know how many there are there. Lots of myofibrils. They're all getting shorter, so the whole the whole muscle fiber gets shorter. So these thin filaments of actin protein and the thick filaments of myosin protein, these are called contractile in a cell. They're the ones that cause it to contract and get shorter. We'll still need to see, how does that happen? How do these overlap more? Pull the sarcomere shorter. We'll see that in a minute. So more terms that we need to learn about the structure of a muscle fiber. We already said the sarcolemma is just a special name for the plasma membrane of a muscle fiber. Just a membrane that covers the whole muscle cell. Transverse tubules, or T-tubules, are little tunnels that they are part of the sarcolemma. So that's what we see here. There's the sarcolemma. That's just the plasma membrane covering the whole muscle cell, and we see those T-tubules are just tunnels that are extensions of that sarcolemma that go down deep inside the cell, and we'll see what's their function in a minute. The cytoplasm, that is the inside part of a muscle fiber, also has its own name called a sarcoplasm. In the sarcoplasm, there's always a lot of glycogen. Remember, glycogen is a chain of glucose molecules. We store glucose in our bodies as these glycogen molecules. If you got more glucose in your blood than you need, that glucose will be put together into long chains called glycogen and saved for later. And we save glycogen in our livers and also in our muscle tissue, because your muscles use a lot of glucose for energy, for making ATP, and the ATP provides the energy for doing this. We'll see how. There's also this stuff called myoglobin stored in the inside of muscle cells. Probably know about hemoglobin. Hemoglobin is this is the red stuff in blood. Hemoglobin. It's red because it has iron in it. But hemoglobin is just a protein molecule in red blood cells. What's the function of red blood cells? Carrying oxygen. And the oxygen attaches to the hemoglobin. So it's like when we inhale oxygen, the oxygen goes into our blood, and then the oxygen attaches and hitchhikes on the hemoglobin while it's in your blood. In your muscle tissue has its own protein similar to hemoglobin, but it's called myoglobin. Myo means muscle. Hemo means blood. So same kind of thing. It stores oxygen in your muscle tissues because your muscles are constantly breaking down a lot of glucose for energy, and they need oxygen to do that. That's cellular respiration reactions. So your blood is always delivering oxygen to your muscles, but your muscles use so much oxygen when you're using your muscles that they store their own oxygen in this myoglobin. Yeah. There's also a special endoplasmic reticulum in muscle cells. Remember, we learned the endoplasmic reticulum in cells is for making proteins and lipids. But in muscle tissue, there's a special kind of endoplasmic reticulum called the sarcoplasmic reticulum, and it has an extra function of storing calcium, stores calcium ions in there. And we'll see why that's important in a minute. So that's what's shown here, the sarcoplasmic reticulum. These hollow branching tubes that hold a lot of calcium. I already said some of this stuff: myofibrils, sarcoplasmic reticulum, cisterns, terminal cisterns are. A cistern is like a tank, and so that's what this is one of those terminal cisterns. It's like a tank that stores a lot of calcium. That's important for muscle fibers to contract, and we'll see how that happens in a minute. Don't worry about that word triad. We don't need to know that. Okay. Here again, another view we see. That's a muscle fiber there, and myofibrils. See, each myofibril has a sarcoplasmic reticulum wrapped around it that holds a lot of calcium ions. Here's a section of a myofibril, which is a sarcomere. You see the thick and thin protein filaments. Here's an enlarged view of thick filaments and thin filaments that are within a sarcomere. And we see again they overlap. The thick filaments and the thin filaments are overlapping. But in this view, we see these little they're called heads or cross bridges sticking out of the thick filaments, and some of them are attached to the thin filaments. These are called myelin heads, myosin heads. These long myosin protein molecules have these hooks on the end that stick out. They're called myosin heads or cross bridges because they attach to the thin filaments. Again, same thing, enlarged. We see these long myosin protein molecules, and each one has a hook on the end sticking out. You see some of them are attached to the thin filaments. We said the myosin and actin of the thick and thin filaments are called the contractile proteins. But there are also a couple of kinds of proteins that are called regulatory proteins that it switches the contraction on and off. We'll see that happens in a minute. There are several other proteins that are called structural proteins that we don't need to learn the names of them, but they just make up other parts of this. Like where the Z disc is, that's made up of a certain kind of protein molecule. There's other protein molecules that attach the thin filaments to the Z disc, so on. Those are all called structural proteins. They just hold this whole arrangement together. This one, here's another one of those structural proteins here. We don't need to learn the details of all of that stuff. But here we see again, this is an enlarged view of a thick filament. So these are all these long myosin protein molecules with their heads sticking outside to the side. There's an enlarged uvula, one of those myosin molecules, head sticking out. And here, this is a thin filament. So this is a thick filament. This is one of the thin filaments. And it's made up of these protein molecules called actin that are shown as these yellow balls. Yes. These are the actin protein molecules that make up this thin filament. But we also see two other kind of proteins on here. This one that kind of twists around the thin filament or twists around the actin is called tropomyosin. That's one of those regulatory proteins. The other regulatory protein is troponin, and it's shown as these looks blue to me, these blue football-shaped things. And on these actin protein molecules, you see each one has a dark spot on it. Those are called the binding sites. Binding sites or the binding locations, because that's where these myosin heads from the thick filament can attach. And they can reach up and attach onto the binding site of one of these actin molecules. And that's what we see. That's what we were seeing here. This is where some of these myosin heads had reached up and hooked on, attached to the binding site on some of these actin protein molecules on that thin film. When they do that, it's called a cross bridge. Cross bridge of the myosin attaching to the actin. Thick and thin filaments are attached together there. When the muscle is not contracting, this tropomyosin protein molecule is covering the binding sites on the actin so that the myosin heads cannot attach. But when the muscle is going to contract, something that has to happen is this tropomyosin has to move out of the way to uncover the binding sites on the actin to allow the myosin heads to attach. Myosin heads can't attach, and it can't attach. What causes the tropomyosin to move off of the binding sites when these troponin protein molecules change shape? They change shape and they pull the tropomyosin off of the binding sites on the actin of the thin filaments so that the myosin heads can attach. That's why they're called the regulatory proteins, troponin and tropomyosin. They regulate when the actin binding sites are covered or uncovered, so that the muscle fiber can either not contract or it can contract. Okay, here again we see a different kind of diagram showing that the binding sites on the actin, binding sites for myosin on the actin molecule, normally they're covered by this tropomyosin molecule. But this troponin is attached to the tropomyosin. And when this troponin changes shape, it'll pull the tropomyosin off through the binding. This troponin, anybody know what it means if a patient has troponin in blood? A patient shows up at the hospital, experiencing some kind of medical condition, and one thing that they would check for is troponin in the patient's blood. You know what that condition would be? A heart attack. Because normally you don't have this in your blood, but if you have a heart attack, that means part of your heart muscle is dying, breaking down, and now you got this protein from your heart muscle breaking down. Now you got troponin in your blood. That's one sign of a heart attack. You should have troponin in his blood. Same thing could happen if some patient has some kind of traumatic injury to a muscle. You could end up with troponin in the blood. So it doesn't always mean a heart attack. Okay, well, so what is it that causes these troponin molecules to change shape and pull the tropomyosin off the actin binding sites? Calcium ions. That's the calcium that's stored in the sarcoplasmic reticulum until the muscle is going to contract. Then that calcium is released, it comes out, calcium comes in contact with it, causes it to change shape, pulls it off pulls the tropomyosin off the actin binding sites. Now the cross bridges can attach. Okay, these structural proteins, alpha-actinin, titin, nebulin, myomycin, dystrophin. Might be a question on the exam which would just say, just list all these and say what are they. And all you'd have to know is that they're structural proteins. You don't have to know what the different functions are or where they are. You just should recognize. If I give you all those words, you should recognize, yeah, those are the structural proteins. But that's all you need to know about them. Okay, here's showing a diagram of a muscle fiber. Actually, they're very long like spaghetti, but they're just shown kind of square here, just to simplify it. And so these myofibers are actually very long. There's a lot of them inside one muscle fiber, but there's only showing a couple of them. There's a mitochondria that are making ATP that are used for the energy to shorten these myofibrils. But and the whole thing is covered by the sarcolemma, that is just the plasma membrane surrounding this whole cell. And there we see the T tubules, transverse tubules, these tunnels. They're just continuations of the sarcolemma that go deep into the cell. And they come right next to those cisterns of the sarcoplasmic reticulum, and those dots in there, those are all calcium ions. They're stored in that sarcoplasmic reticulum. So when this muscle needs to contract, the calcium will be released out of there, and the calcium will come down into these myofibrils, where the troponin is covering the troponin and the tropomyosin covering the binding sites. We still need to see how that happens, what causes it to happen. Case neuromuscular junction. Neuro means nervous system. Muscular, of course, refers to muscle. So a junction is a connection between two things. A neuromuscular junction is where a neuron, a nerve cell, meets a muscle fiber. If I decide to want to contract this muscle, there's neurons going from my brain to that muscle, where a neuron meets the muscle fiber that I want to contract. That's a neuromuscular junction. The muscles that are the neurons that carry voluntary information from your brain to a muscle are called somatic motor neurons. Somatic means voluntary. So anytime you decide you want to contract a muscle, that information will go from your brain through a somatic neuron to those muscles. For that to happen. The connection at the neuromuscular junction is called a synapse. That word, we also use that word where two neurons come together. The end of a neuron is called the axon terminal. The word terminal just means end. The axon is that long extension of a neuron. Oh, need to look at a diagram showing. Um, well, so here would be a neuron. Uh, a neuron looks like a couple of hot dogs. At the end, each one of those branches has an axon terminal at the end, where it forms the neuromuscular junction with the uh, muscle fiber. Um, so the nerve impulse go through that neuron, and when it gets to the muscle fiber, it's going to tell that muscle fiber to contract. How to do that? Here's an enlarged view of that axon terminal, the end of the neuron, and this is the surface of the muscle fiber. When a nerve impulse comes along that neuron, gets to this axon terminal, there are these vesicles in here containing molecules called neurotransmitters. Neurotransmitter, in this case, the neurotransmitter is called acetylcholine. Um, ACH is short for acetylcholine. That's what these are, acetylcholine molecules. And the nerve impulse causes these vesicles to move to the surface of this neuron, this axon terminal of the neuron, and release the neurotransmitters into this little gap here. So the little gap between the end of the neuron and the surface of the muscle fiber. This little gap is called the synaptic cleft. This is a synapse here. This gap is called the synaptic cleft. So when these acetylcholine molecules, they just drift across the gap and they come into contact with receptors on the muscle fiber. These receptors are actually nanoproteins. That when this acetylcholine comes into contact with this receptor, it causes this tunnel to open and allows sodium ions to come in here, positive sodium ions, and that changes the electrical charge of the um, sarcolemma, the plasma membrane. And then from there, it's like a nerve impulse, but it's on muscle that'll spread out over the whole surface of the muscle fiber when this happens at the neuromuscular junction. So this is like this electrical charge, this electrical change um, impulse spreads out over the entire sarcolemma. Go back here. This whole sarcolemma covering the whole cell, it'll have this impulse, electrical impulse spreading out over it. And when that electrical impulse goes down the transverse tubule, which is right next to these cisterns or these tanks of the sarcoplasmic reticulum that contain those calcium ions, it causes the calcium ions to be released. Calcium ions come flooding through all of this, and they go in here and cause the troponin molecules to change shape and pull the tropomyosin off the binding sites of the actin molecules. Anybody still awake? I know this is hard to hear. It's just as hard to explain it. You need to learn this. You need to learn what all these steps, what's going on here. It all makes sense if you learn it and understand it. So the acetylcholine is rapidly broken down. So there's an enzyme there at the neuromuscular junction. So this acetylcholine neurotransmitter molecules that are being released here, they don't just accumulate or stay there forever. soon as they stimulate, cause these channels to open up and allow the sodium to come in to cause that impulse that spreads out over the muscle fiber. There's an enzyme that'll break down these neurotransmitter molecules so that the muscle will be ready to go again. It won't just stay stimulated forever. Okay, here's an actual electron microscope picture of a neuromuscular junction. So these are the neuron, and it branches out into these several different axon terminals. Each one will form a neuromuscular junction with on the surface of these muscle fibers. Here's another view showing, summarizing all this stuff. So here's a neuromuscular junction. Nerve impulse comes down there, causes the acetylcholine neurotransmitter to be released into the gap, synaptic cleft. When they contact the receptors on the surface of the muscle fiber, it causes this impulse that spreads out over the whole muscle fiber. It goes down those transverse tubules, causes calcium to be released from the cisterns of the sarcoplasmic reticulum. This calcium ions come flooding through all of this stuff. When these calcium comes into contact with these troponin molecules, they change shape and pull the tropomyosin, these long twisted ones, pull them off of the binding sites on these troponin, or these actin molecules of the thin filaments. That allows the myosin heads to reach up and hook on to those binding sites on the thin filaments. So up here, the binding sites are covered, so the myosin heads are not attached. But out here, you see that they're attaching. So once these myosin heads or cross bridges attach to the thin filaments, then these cross bridges they bend like this, and they pull the thin filaments toward each other. Each one of these myosin heads will reach up, hook on, pull, and then they'll release, and other ones will be hooking on and pulling. And so there'll just be this pulling, as these thin filaments overlap the thick filaments more and more and more. The whole sarcomere here gets shorter and shorter. All these sarcomeres doing the same thing, so the whole myofibril gets shorter. And what's providing the energy for these cross bridge do that? ATP. So every time a cross bridge hooks on and cranks like that, it uses one ATP molecule. Hey, this so-called excitation contraction coupling, that's just what I've been describing. The nerve impulse comes down the neuron, gets to the neuromuscular junction, stimulates or causes this whole impulse to spread out over the muscle, and causes it to contract. Causes it to contract by causing the calcium to be released, which moves the tropomyosin off the binding sites, allows the myosin heads to attach. And another diagram showing this. Here the tropomyosin is blocking the binding sites. Here calcium has caused the troponin to change shape, pulling the tropomyosin off the binding sites. Now the binding sites are exposed, so the myosin heads from the thick filaments can reach down and on, and then each one of these cross bridges will pull this way and pull the thin filaments toward each other, and the whole sarcomere gets shorter. This is called the sliding filament mechanism, because the thick and thin filaments are sliding past each other, actually being pulled past each other by the cross bridges. Here again we see another diagram. See the thin filaments here are being pulled toward each other. Here the sarcomere is all the way shortened. The thin filaments are actually running into each other. So it can't really get much shorter than that. Okay, so a list of the steps involved. ATP hydrolysis. Hydrolysis just means that the ATP is being broken down. That third phosphate is released, so the energy is removed, so the energy is released, and that energy is used for the cross bridges. Attachment of the myosin head to the actin of the thin filament, and then the power stroke. The actin head is just standing like this, pulling on the thin filaments, and then in order for a cross bridge to detach from the actin binding site, an ATP molecule has to be there. It won't release until a new ATP molecule is there to cause it to release, and then it causes it to release, straighten out again, and hook on again, and crank. And it won't release again until there's another ATP molecule that'll cause it to release, straighten out, hook on, crank. Another ATP molecule. Release, straighten out, crank. So here we see that the ATP molecule has to be there right at that cross bridge in order for it to release and crank again. So is yet another set of diagrams showing the same thing in a little different way. So neuromuscular junction, the impulse going down the transverse tubule, causing calcium to be released from the sarcoplasmic reticulum. All these calcium ions flood into the sarcomeres, cause the tryptomyosin to move off the binding sites on the thin filaments, and all these myofibrils get shorter, so the whole muscle fiber gets shorter. Let's stop here. That's enough. We'll have time next week to finish this chapter and go over the list of muscles that we need to learn.